Okay, thanks everybody. It's nice to be here. So my name is Todd Taylor, and as Guillaume said, I've at RIKEN. I've been there for longer than all of you have been alive. I won't say how much longer than that, but obviously I'm not Japanese. I come from the U.S. Seattle area, but I've been in Japan about 23 years or so. So I had a team at RIKEN. Some of you have been to RIKEN. So it's one of the longest running and largest basically government-funded research institutes in Japan. It's been around for 100 years. We're going to have an open day at the Yokohama campus, and you're all welcome to come. So that day it's nice where you can go into the campus and you can go around and see what's going on in different labs, and there's a lot of fun events and things. So I had a team for the past 15 years, a bioinformatics team, and just last year I changed my position slightly to I'm a coordinator for our center. We have a lot of international collaborations going on. Our Center for Integrative Medical Sciences has over 400 people, 54 labs, doing research in the areas like immunology, genomic medicine, sequencing, and developing a lot of new technologies for sequencing and other DNA related things. And I'm also a senior research scientist in the lab for microbiome sciences. So do you know what the microbiome is? Maybe not. So in, on, and around us, there are thousands, millions, billions, trillions of bacteria and other small microbes. So the human, our genetic code, we are not alone. We are actually sharing our environment with all these bacteria, viruses, small other creatures, but they are actually, we are living together in harmony in most of the cases. So that's the kind of thing that we're studying. Sometimes things go wrong, and some bad bacteria can take over in different parts of your body, leading to different diseases and things. But for the most part, they're there to protect us. We give them a nice, warm environment to live in. So that's the kind of thing I've been interested in for the past 8 years or so. So just a quick background. So again, I've been at RIKEN 20 years, and I started there in '98. I was working on the Human Genome Project. So you've heard of the Human Genome? Okay. Whew. Okay. So that was really exciting times. That's the thing I wanted to do after graduate school, because that Human Genome Project had just kicked off. And RIKEN was the major player in Japan. So this was a large international project. But we sequenced part of 3 chromosomes, 11, 21, and 18. And then after that finished, we did for a few years, we did some comparative genomics. We sequenced a few of the chimpanzee chromosomes to compare, try to get some idea how different we are from our closest evolutionary cousin. And then for the past 10 years or so, I've been working again on these microbiome studies, mostly on looking at microbes in the skin, like the oral cavity, and the gut, which usually come from fecal samples. Okay, so that's my history. So now I'm going to give you all kind of a quick introduction to big data, biocuration, and annotation. So just quickly raise your hand if you've heard sometimes in the news recently about big data. Okay, a couple people. So now, as you know, there's lots of information on the internet. So by kind of running some statistics analyses over the data, we can start to make some predictions. So for example, Amazon, if we look at their purchasing history, we can see, maybe there's a cold or a flu coming through Japan because suddenly all these people are buying all these cough medicines and other things like that. And we upload lots of information. We do Google searches, and we upload pictures on Facebook and Instagram, and people are always kind of analyzing these things, and they make predictions for you. If you ever noticed in your email software, they might say, are you interested in such and such project or products? That's because they're kind of scanning your emails and other information, say, he or she likes these kinds of things, so they suggest. That's just kind of an example of a big data. So biocuration is the next thing. So bio, that's the biology part, and curation, that's kind of like adding information. So we want to add information to some of these data sets that we're generating to make them more useful. And again, I'll give you some examples here. If you have any question anytime, please feel free to interrupt or to ask me. You're not interrupting, just raise your hand and let me know. If you're confused, I'd rather explain it than just keep on going. So if you go to Wikipedia, you all know Wikipedia? Okay, whew, that's another one. Okay, if you don't know that, I'm in trouble. So there's just one page where they talk about information collections. So I've just kind of highlighted the ones that have to do with biology. So I'm not trying to discriminate against the physicists in the group, but the things I'm talking about can be applied in that field as well, not just in biology. But my background is more biology, so I'll be giving examples in that area. So we have like the genomic information, sequence data, lots of papers, publications from all these research and so on. Images, proteins, all kinds of information that's being nowadays generated at really high throughput. And these are just various types of big data sets. Other things are like music, things from chemistry. Oh, I don't know where I'm pointing here. Images, so on. So for big data, there's many challenges. So one of them, or some of them, are like capturing the data. Like how can we accumulate all this information in one place to try to study it? Data storage. It used to be that I couldn't even put the human genome sequence on my laptop because the hard disk wasn't big enough. But of course now, I mean, that's hard disk sequencing, our computer processing, everything has gotten so much better. So now we can store lots of information even on our small device, even on our, our iPhone is more powerful than the computer that was used to get man to the moon way back when. So like everything is, faster, bigger, more, whether it's DNA sequencers or computer processing power. But data storage actually is kind of a new bottleneck. It's becoming very expensive. So if we're sequencing, so when I worked in the human genome, that was one genome sequence. About 3 billion bases of DNA, ACGT, cost about $3 billion for the first genome. That's similar to the cost of taking man to the moon. Now you can sequence your genome for maybe less than $1,000. So because during this past 20 years, the technologies, everything has just dramatically improved, more high throughput, costs have come down, and we have that first reference genome that we created that it makes it much easier for people to resequence the genome nowadays, because you can map all these small pieces of DNA to this big, well-annotated reference genome. Searching through, so we're creating so many large data sets, it's just leading to more and more complications and the greater need for people like who can do bioinformatics, computer programming, analysis and things. So actually creating the data is no longer the bottleneck because that's, again, faster and cheaper, but analyzing the data is the new bottleneck. Sharing information, let's say I'm running a sequencing lab and I want to send the data to you at your lab. The data is too big to send over the internet anymore. Sometimes it's faster just to put it on a hard disk and send it by FedEx or some other Takubin. Visualizing the data, sometimes the data is so large that we just can't look through, Excel sheets, thousands, millions of rows and columns and try to make any sense out of that. So people are working on different kinds of visualization tools so that we can look at these data sets in a visual way, which is much easier for us to comprehend. And trying to keep information up to date. And information privacy is a big issue. Another big data format that's talked about in recent years is hospital, like patient data. So all these hospitals are have been collecting data on their patients, of course, for a long, long time. Each hospital has its own particular system. There's no common, or there hasn't been any like common way that they've been storing and collecting this information. But again, we have so much data collected there. If we could kind of bring that together and analyze it, I'm sure there's a lot of things that we might be able to discover from that information by comparing large data sets. But we have to be careful to keep people's privacy. We don't want to reveal the names of the people, of the data that we're looking at. So we have to be careful how we store and transmit the data and share with other people. And so the traditional web, and I know you're all familiar with the web, is basically a web of documents in many, many different formats. So we've all heard of HTML, kind of the basic formatting for web pages. So it makes it very easy for humans to read. But if we're trying to extract information from those pages, it's not so easy for computer algorithms to scan through the pages, because website A might store the data differently than website B does, than website C, and so on, because each of them is using the format that they're comfortable with. So this is kind of the traditional web of documents that are not necessarily linked or not easily linked to one another. The semantic web is a web of human and machine-readable content. So from the human's perspective, the page probably still looks the same, but now in the underlying, in the HTML or XML, the formatting used to create the pages, they're using common tags. Let's say I have 5 different sites that are talking about genes or something. Rather than just putting the name of the gene, invisible to the human eye because it's hidden in the HTML, they're using some kind of tag which says gene ID name. And each one of these sites is using the same tags. So if we can all share these tags or kind of common vocabularies, the way that we mark up the information in the web pages, now we make it easier for computer algorithms to like crawl through the web pages and extract the same information from different pages because of this tagging, this controlled vocabulary system. So the semantic web it gives a common framework, and it allows data to be shared and reused across applications. And it's a web that can be processed by machines. So and much of it is machine readable. So RDF, so Resource Description Framework, this is a way that we can, again, express information by using certain formatting and certain tagging that for the human it's not so easy to read. And again, that's probably hidden in the web page, but it makes it easy for computer algorithms to crawl through. So the resource can be anything. It can be a document, person, physical object, and so on. And they use this called triple format, so subject, predicate, object. So it's simply like author's name is Bob. Okay, so subject, predicate. If you do this same kind of formatting for the information in the pages, now we start to be able to link things together. So for example, Bob, I just made up Bob, totally, and I didn't realize the next slide was going to have Bob in there. So, okay, it must be a common. So for example, these triplets, like Bob is a friend of Alice. Bob is a person. Bob is born on July 14th, 1990. Bob is interested in Mona Lisa. See, these are all triplets, subject, predicate, object. La Joconde, I shouldn't read this, I should have teach us French, okay. La Joconde Washington, okay, no. Is about the Mona Lisa. Okay, Mona Lisa was created by Leonardo da Vinci. So now you can see I've kind of made a graph out of the web. So now I can actually connect Alice to Leonardo da Vinci through, if I want to, like, query. So, and you could ask some, really weird question like, of Leonardo da Vinci's paintings, is there anyone named Alice who likes these or something? So we can start to ask questions based on this linkage. And then sometimes we have what's called like a blank node, where the background includes just this area, and in this area is a cypress tree. So this is a little technical there, but so sometimes the nodes are actual objects themselves, and sometimes they're just nothing, or kind of referencing a space. Okay, so that was a quick introduction to kind of semantics, and now I'm going to talk just a little bit about biocuration. So biocuration involves, I don't like to read my slides. I don't like when people read their slides, but I'm going to read my slides in a few cases, so please don't give me low marks for that. But don't ever read your slides. So it involves the interpretation and integration of information relevant to biology or physics or whatever field into a database or resource that enables integration of the scientific literature as well as large data sets. So primary goals are to create accurate and comprehensive representation of biological or of knowledge. I'll try to not say biological so much. And then make it easy for scientists to access this data so that they can do analyses over it. So if I have 100 research papers about the human genome, all written by all different authors and so on, but I want to go through that and I want to pick out anytime they reference a gene name or a protein, or if there's some interaction, protein A causes this disease, or protein B interacts with protein C, and then lead. Those are the kind of things we want to extract from all the scientific literature. But the only way we can really do that is for a human to read through all these papers. And because we want the most reliable information from the paper. So this is the kind of the jobs of biocurators. It's a very time-consuming process, but the end result, it means we have highly accurate and reliable information. People are trying to develop computer algorithms to scan through these papers and pull out the same kind of information, but it's not always easy because we can write things in many different styles. A lot of genes have alias names and things like that. Sometimes the gene name can almost look just like a word, so it doesn't necessarily just pop out, oh, this is a gene name. Unless in the background there's some of these tags. So if you look in Wikipedia, this is a biocurator is a person who curates, collects, annotates, and validates information disseminated by biological and model organism databases. So we have the human genome, we have mouse, rat, worm, fly, and so on. All these people in those fields are going through, anytime a new paper is published, they look through these papers and try to pick out the kind of relevant, the key information, and then put that into their larger database so that the people who are working on those organisms, they have it all in one easy to access location. Again, in a format that's going to be much easier for a computer to analyze and to process and search through. So their role is to basically quality control of extracting that information, or even to work with people sometimes before they publish their papers so that they use the proper kind of tagging or labeling to make it easier for other people to extract important information. I've already said that. And so they want to describe the data using standard names, standard annotations, protocols, vocabularies. And then by standardizing the way that we describe the information, and that just makes all the different data sets work together more friendly. And sometimes they have to contact the researcher to see, what did you really mean by this particular information, if they really get stuck on trying to extract it for themselves. So sometimes they're called scientific curators, data curators, annotators, data wranglers, and so on. But probably the most common one is biocurator. So this is just an example. You don't have to know any details of any of this, and the slides will be available, I believe, later. I've given them. You can always email me and ask if you had some specific question too. There's a very famous, protein database where they want to have the most reliable information about all of the known proteins. And so what they do is they have to go through all these other different resources, look through sequence databases, look at sequence variation, protein name information, look at protein functional domains, look through the literature, maybe some of the papers that have been referenced, that have referenced the particular protein that they're interested in. And these are these kind of common vocabularies, also called ontologies, where we try to use standardization, the way that we label information within the paper. So the curator is this person here who has to basically, in order to extract the information out of here about their protein, they've got to go to all these other these other resources to try to find the best information they can. And there are, I think in biology, I've seen a few years ago that there's over 1,500, maybe 2,000 different, at least biological databases. Each one has different types of information, but also large overlaps. So it doesn't matter what field you're in, but there's not just a one go-to place for any of this. So ontologies, so these are these controlled vocabularies I talked about. So it's a way of formally naming and defining something. So when we're talking about some entity, something, we want to know what kind of type it is, properties about that, and how does it interrelate with other things. So this is just an example. It's called the gene ontology. It's probably too small to read, but basically at the top here, so it just says biological process, and then there's subcategories of that. So cellular processes, signaling, biological regulation, and then each of these can break down into further. So you can go down into the very fine, specific information about what a gene does, if that information is known, in this very hierarchical. So all of these functions and these terms are these standard vocabularies that they encourage researchers to use when they are describing their data. So if we're all using these same kind of words, terms in our papers, in our databases, again makes it easiest for us to integrate the data from database to database. So this is just another way to look at the gene ontology. So the same information that's here, you can just see in this kind of tree view. So here's biological process, cellular component, molecular function. And then molecular function is broken down into all these other things. If you're not a biologist, don't worry. Even if you are a biologist, you probably don't know all of these. But some of them break down into more and more subcategories, getting into very specific features. So this is just kind of what a standard ontology page looks like. It doesn't matter what the subject is, but they'll give you all of these terms which are linked, and then there's more information, maybe some descriptive information about each of these as you click on and expand them. I will be using this a little bit later today, so this ontology lookup service. So again, when we describe information, we're going to be looking at some scientific papers, and I'm hoping I can get you all to make some small contribution if everything goes well here. But this is just one place one resource where you can go and you can, let's say I'm looking for the term just even language. So I would type in their language, and it might give me several options, and some with different descriptions. I'd have to pick out one, or let's say I looked for English. So that could be a language, or it could be a person's nationality. So you want to choose the one that has the most appropriate definition. And there's many different ones. I mean, some of them have to do with physics, mathematics, chemistry, ancestry, taxonomy, and so on. So people have, communities work together to develop these vocabularies and then share those with everybody, encourage people to reuse, don't recreate. Because every time we recreate a new vocabulary, that basically separates our information from all the rest. So just like with recycling, we should try to reuse terms and things that are already there. So when we do that, that makes our data connect with other people's data. Have you heard of crowdsourcing? Crowdsourcing? So there's different things. There's crowdsourcing, crowdfunding, and most of them are through using the internet. Something almost anybody can participate in. So whether it's individual, institute, company, or so on, with varying degrees of knowledge. And so basically in any kind of crowdsourcing or crowdfunding, they're looking for people to volunteer their time or volunteer some money to a larger project. Something that couldn't be done by a single or small group of people, but you need hundreds or thousands of people contributing. So this is something where the crowd should participate. So, and then you bring your either money, knowledge, experience, and so on into the project. Sometimes you don't need to have really any prior experience. You can learn like at the time that you volunteer to do this. It's just something that sounds interesting to you. And we hope that in these kind of crowdsourcing activities, the hardest part in a crowdsourcing thing is to get the crowd. You know, everybody's so busy doing their own thing. You know, so why do you want to volunteer some of your precious time to my project when you already have all of your own projects and things? Sometimes in a crowdsourcing project, we can give some financial support, some small amount for contributing. But the main thing we're hoping is that you'll feel good about yourself, that I contributed to this resource, which now I can see is being useful to the rest of the community, to the rest of society. But can help you as well. Can somebody give me one example of the most probably well-known crowdsourcing thing on the internet? Louder? Wikipedia? Wikipedia? Is that what you? Yeah. So has anybody here ever contributed to Wikipedia? Okay, a few. So how many of you have used Wikipedia? Oh, okay, now. So for all of you who haven't contributed, do you sometimes find information that you're looking for is missing? Yeah, so I contribute now and then, not really in the field of like biology, but to my favorite artists or authors and things. So sometimes I go in there and I want to see what is their latest album or book, and it's not there, but I know it's coming out, so I'll go and I'll add it to the page. And now that information is there for everybody to share. So anybody can contribute to Wikipedia. There's always going to be some problem that maybe some people try to put some like bad language or images or things like that. But nowadays there's a lot of quality control, and people check out the pages and they look for anything that seems strange and they just remove it immediately. But yeah, this is again, if you go on there and you're looking for some information that you know should be there but it's not, then you're more than welcome to add it. And then the next person that comes along doesn't have to kind of waste the time that you did. So there's many different crowdsourcing things. Again, too small here, but some of them is like Internet Movie Database, One of my favorite ones, not crowdsourcing, but it's crowdfunding. Have you heard of Kickstarter? Okay. Has anybody here ever played Baku Hatsuneko? Exploding Kittens? Okay, it's a card game. It was the most well-funded Kickstarter game. So that's called crowdfunding. So crowd, again, many people. So let's say I have an idea for a project. In this case, they had an idea for a card game where, if you picked up the cat that was exploding, you lose the game. And everybody thought, wow, that's a great idea. They only needed about $10,000, $20,000. If they get the money, people contribute $10 here, $20, $50. If they get enough money, they are obligated to make the product and then give you whatever your reward is. But in their case, even though they only hoped for $10,000 or $20,000, they got over $2 million. So, because people thought, wow, this seems like a really fun game. And if you've never played, you should play. I don't make any profit off of sales of this game, so we play it at work sometimes on a Friday evening. So. There's many kind of these crowd activities. Again, you can't do it on your own. It requires a lot of people, or it requires either people investing their time or maybe their money. And then, like anything, a little bit adds up into something much larger. Okay, so now I'm going to introduce you to a tool that we developed in my former team that we're still working on. And this is a crowdsourcing biocuration tool. See how it's all coming together? So I talked about biocuration, crowdsourcing, and big data. So just the kind of title of this second part is like turning big data, which when I talk about big data, other people might have a different definition. Mine means basically kind of unstructured data, like the web pages I talked about or scientific papers. There's no common structure from one thing to the next. So into small data, now small data will be extracting that important information and putting it into a database, which makes it become searchable, analyzable, and so on. So that's my turning big data into small data through crowdsourced curation. So integrating all types of medical and scientific knowledge. So I've already mentioned that there's so many things on the internet now. It doesn't matter what field you're in, whether it's science, chemistry, physics, mathematics. There's so many hundreds or thousands of papers that are being published each year. And so much data in your field is, again, being generated, put up on the web for people to use and take advantage of. But it's too overwhelming. So we need somehow to kind of manage all of this. So this is my kind of former team. So we were very international. So we have an American, French guy. You always got to have one French guy. Indian. He still works with me in my new team. Korean and Japanese. So the name of our tool, it's called iClickVal. So this is my own original logo. So it looks like somebody's clicking. So I click on something and give it value. Okay, but I'll explain more later. So those are the two main guys that were developing this tool with me. So you're still a bit young, but I'm sure you've had to write some papers in your time already. And so the first thing that at least we do in our field, if we're going to write a paper or a grant, we would go, in biology, we go to this database called PubMed. Has anybody heard of PubMed? Maybe some of the people in the biology field. So again, I'm just going to be giving the biological backgrounds, but I guess if you're in physics or chemistry, mathematics, you might have other similar types of resources that you would go to search through the scientific papers. So, but for us, it's basically this PubMed. And it's a free search engine that's supported by the NIH in America, where they have a huge database of references and abstracts about papers from the biomedical field. Not the full text information of the paper, just the title, the abstract, and then some of the, like the author information and some other key information. But and again, I'm going to give some more examples about microbiome or metagenomics, metagenomic field. It's the same thing basically as microbiome. So I'll use that in my examples. So if I searched in the PubMed database for the term metagenomics, I get like over 5,000 papers that give me back some hit. So that's plenty, you know. I mean, do I have to look through all those 5,000? That's not a very specific search term. Another place, I could go to Google Scholar, and maybe some of you are more familiar with that. That's a little bit similar, but this, they've collected and scanned papers from a wider range of fields, not just in biology. If I were to search metagenomics in their database, I get over 40,000 hits. Okay. Or if I went straight to Google, I'd get over 1.5 million. And of course, I'd get a whole mixture of different kinds of things, research papers, wiki pages, videos, and so on. And so I need to give more specific search terms to kind of narrow down what I'm looking for. But basically what I'm trying to say is searching for scientific media or searching media just in general is not trivial. We don't just go to Google and that gives us everything we want, but usually it gives us so much that we don't bother to go and look other places. The problem with that is that we might be missing critical things just because we didn't use the right search terms. And most things are somehow in Google somewhere because they're going through and they're crawling through all the web pages and scanning everything. But again, it's like so much information, we're not maybe getting into the thing that we're really looking for. So there's no, I said, single kind of go-to site. So I want to, it doesn't matter what field you're in, I want to have one place where I can go to and I can put in a very specific kind of search. I can say like, hey Siri, give me all the papers on the gut metagenome that have been published since 2009 that include an image in the document. So that's my kind of dream question that we can do with our database. I can't do that today, but this is the thing we're kind of shooting for. If I were to ask Siri that question right now, she would probably give me some answer like, you know, does not compute or something. So definitely would confuse her. So we don't realize it in the, again, whatever fields you're in, that every day we're basically doing some kind of curation. We're adding some information to something, some kind of media thing, but we don't really know that we're doing it. So for example, I have like hundreds or thousands of bookmarks in my browser. And I've got them categorized by, these are all like research projects, metagenomics, R16S RNA sequencing data, analysis tools, and so on. I don't know if you're like me, but I always like, oh, I'm going to make this bookmark, I'm going to put it here, and I'm going to come back to it later. I never ever go back through, and it's easier just to search the web again and like find the site. Maybe this is dead, maybe now I've kept it there so long the link is dead. So, but you can see I've actually added some information to these links. I said all of these pages have something to do with this taxonomic classification. And all of the ones in this folder have something to do with sequencing methods and so on. So I've basically simply said this site has something to do with this subject. So that's a type of curation, but it's just private to me. You know, I should be able to share this with people so that we're not all wasting our time. I have like hundreds or thousands of PDF files downloaded, again, categorized by if they're on the human genome or metagenomics or so on. You know, they're all labeled with this generic 1, 2, 3, 4 kind of label. I'd have to open it up again to see what was actually in there. I know it has something to do with this subject in my folder. So again, I've given a little bit of information. Or I might print out a bunch of papers and put them in binders and say these ones have to do with genetics and this one has to do with physics and chemistry and so on. You know, stack them up, never read them. Maybe you have a bit more time on your hands, and somebody a few years ago asked me to write some paper about all of the tools that we can use to analyze this microbial, this metagenomic DNA. So I had to go through and I had to search PubMed and online using all kinds of different keywords and things. And I started making this huge Excel table that had the title of the paper, and I added in a column or two with keywords, so I could say this paper has to do with this subject and so on. Again, I'm adding some information to that paper, but it's just private to me again. So, and I'm just one researcher out of millions around the world, but we're doing the same thing over and over and over again on the same media. We're looking for it, we're labeling it, we're extracting the same kind of information. The way that I look at a paper, the way that you look at a paper, another person looks at it, we might be coming from different kind of backgrounds, so we may be looking for different kinds of information in the paper. I might be interested in whatever genes were mentioned in this paper. Another person's interested in what kind of tools were used in the paper. So it doesn't matter what kind of perspective that you're looking at this same thing in. What we want to do is be able to, that doesn't mean I'm right and you're wrong, but if we can gather all of our knowledge together in one place, then we'll have a better understanding of what this is. So if we just communicate with our fellow scientists of what we're all looking, we might understand the larger picture. Oh, okay, this is an elephant, this isn't a rope or a spear or a rug or a snake. So let's use the power of crowdsourcing to share this knowledge. So the kind of information I'm talking about previously here, none of that is like top secret information. I'm looking at it and said this paper has to do with this. If it gave me some idea oh, if I do such and such, I might make some big discovery. I'm not going to share that information with other people, but the basic kind of information that we can extract and see from the paper, why not share that with people and just stop wasting our time? So I've already mentioned PubMed, but also I'm talking about scientific things. So of course we know most of the things on YouTube are not scientific, but there are many scientific videos, including from RIKEN, and even on these other sources. Flickr may not have much of a lifespan anymore. Maybe Instagram is taking over for that, but photo, images, SoundCloud is audio files. Maybe you're not familiar with this, a digital object identifier. So many of the journals for these scientific papers, it doesn't matter what field in, they start to assign a unique ID number for each paper, so that we have an easier way to tell this one ID belongs exactly to this one paper. And there's a lot of these now. This is becoming standardized, not just in biology, but in all areas of science. So I've already talked mostly about these. So PubMed has over 28 million articles or books in its database. There's over 150 million of these DOI objects, mostly scientific papers, theses, data sets, and so on. YouTube, of course, billions of videos, and others. Even Wikipedia has over 43 million pages in about 200 different languages, Internet Archive, and so on. Basically, we're kind of reaching this infinity point of how many different kinds of stable things there are on the internet. Just because I spent so much time looking at this PubMed database, I always find a lot of interesting things there, but basically, as of recently, there's over 29 million papers. Most of those are in English, but only about 75, 80% or so. And then we have about 4 million in non-English. And this is my favorite category, 322,018 undetermined language. So, what does that mean? Is this some I don't know what language this paper is written in. This is what I'm hoping that you can help contribute to a little bit later today. Can we look at these papers and figure out what language they are? So, in 2018, there was over 1.3 million new papers added to this database. So you can see that, again, that's 1.3 million new papers just in 1 year. So this is, again, one of these data resources that's almost exponentially increasing. So that's over 3,600 papers a day. The oldest one goes back to 1809, even though PubMed's only been around from maybe the late 70s. but sometimes they go through older scientific journals and books and scan documents and include those in their database as well. I just went to a celebration in Tokyo a couple weeks ago, Nature magazine, their 150th anniversary, so they've been around a long time. Now when you submit a paper, a research paper, doesn't matter what field, often they will ask you, could you submit 3 to 5 keywords that describe your paper? So my paper has to do with DNA sequencing and the microbiome and the gut. Okay, so some of the papers have keywords, and then some of them have what are called mesh terms, so these medical scientific headings, and these are usually added by the journal. Some have both, and some have neither. So it's very inconsistent from paper to paper. And most of them, or some of them don't even have the abstract. All they have is just the title. So when I search this database, I only get the information basically that's in the title. So if the title's not very descriptive, I probably won't even get that in my match. So usually we think that when you do a search, the shorter your search term is, the more hits you're going to get. And then the longer it is, the fewer hits you're going to get, because now you're searching for something that's more specific. So the more specific something is, the less things you'll find. Sometimes that happens where if I search the PubMed database for metagenomic, I get 10,896 hits. And if I add an S, make it longer, I get about 7,600. So as expected. So I made a more specific, so metagenomic is a subset of metagenomics. So that's why I get more here. If I put quotes around it, why should that matter? Now I get some different number. So what's going on there? So this has something to do with the search functionality in their database. But this other case, I add on an S and I get more terms back. So that doesn't seem to make sense. I should be getting fewer back because now I'm using a more specific term. And if I put quotes around it, I get some other number, smaller, and if I even put a space like meta-genomes or meta-genomes, then I get 36. This is not a subset of this, it's not a subset of that. There's a lot of overlap, but each one of these has its own unique result, a few of its own unique results. So if I really want to get all the papers that have to do with metagenomics, I've got to use all these terms in my search. I can't just I want to just be able to use one term and get all of them back. But if I just use one term, I'm going to be missing things. So I found this is a little outdated now, but if I want to try to get any possible metagenomic-related paper back, I have this list of about 120 terms I have to use. Then I get back a huge number of results. Then I have to kind of go through those 70, 80,000 papers myself and try to determine, yes, this one actually has something to do with the microbiome or metagenomics, or no, this one really doesn't. Even though it uses the word metagenomic, that was just the name of the journal. It was in the metagenomic sciences, something, blah, blah, blah. But the paper was on a different subject, which happens sometimes. Okay. So again, PubMed is just really, it's like a small amount of data there that they've gathered in this one very useful data resource, but that doesn't mean that covers everything. There's a lot of information in books, news articles that comes from other fields, photos, figures, supplemental information, tables, images that are associated with the papers and so on. So how can we turn that big unstructured data into small structured data? So there's the iClick-Val, which I can't really even remember. It's an interactive curation. See, I can't even remember the name of the tool. I made it up several years ago. Literature key value annotation library. So it's easier just to say iClick-Val. So we want to make this single kind of one go-to and curated. So again, that's the thing. it's curated information. A human has looked at it so that it's more reliable. We're not just doing some kind of running a computer algorithm over some papers and trying to extract information and put them in a database. We sometimes do that, but then the human has to look at the information to make sure that's reliable before it goes into our database. So we want to do very comprehensive but also very specific searches and try to make people's work more discoverable. Again, by maybe connecting these terms, so metagenome, metagenomics, with or without the S, microbiome, and so on. So make it easier, because in science, the way that your work is measured is how other people cite your work. So the more highly cited, I looked at Bob's paper, I used Bob's paper, I used his methodology. So the more that your papers get cited, referenced by other people, then that shows that that was very valuable work. If people can't find your paper, how will they ever reference it or use it in their work? Because science is always building from one thing onto another, onto another. So I happen to be very lucky because I was in the Human Genome Project. So the paper, the first paper we published on the human genome has been cited now over, 12 or 13,000 times. So it's one of the, I don't know if it's even in the top 100, but it's pretty close to. So every day it's being cited by some new paper that has used the human genome data in their work. So you want other people to see your work and cite it, and you want to be able to find what you're looking for. What kind of things would I go through to what kind of information could I extract from this paper? So in that kind of triplet format that we talked about earlier, so subject, predicate, object, or key, we also call it key relationship value. So for example, journal title is +1, title is fast and accurate, blah, blah, blah. Number of authors is 4, author 2 full name is Naveen Kumar, corresponding author is Todd Taylor, and blah, 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 blah. So each one of these is a triple in that subject, predicate, object format. When we do that, when we assign these triplets to the information that's in the paper, what we do now, as in the earlier examples, is we start to link this one media thing with other ones. So in the case of the one paper, other ones that I have authored, or the other people in the paper, or if we have referenced the same paper, we've made those connections, like from Alice to Bob to Mona Lisa to that. So by using the common terminologies in our key value triplets, then now we've linked all kinds of media together. And it doesn't matter if it's a paper, a YouTube video, or so on. So that's what our homepage looks like. And like most things, the best way for you to learn is to DIY, do it yourself. And that's what we're going to do in a few minutes. But basically with our tool, we can go through and we can kind of add these triplet pairs to any kind of scientific media that's online. Right now, unfortunately, so I'm sorry, today you guys are all kind of my guinea pigs. So we just made this huge update to our tool, and we just went live maybe about 48 hours ago. So I'm fingers crossed that all goes well, but at least I should get you on there and see if, again, that problem with the 322,018 papers of unknown language, maybe you can help me sort. So that's something that makes me stay awake all night. Like, I can't go to sleep until those 322,018 papers, the language has been sorted out. So for me, one person, I don't want to go through 300,000 articles by myself. But if I can get people to contribute a little bit, like, oh, that was kind of fun. So it's kind of like doing a little bit of research. So just a few days ago, I was at the Sherlock Holmes house in England. And so I feel like a bit of a genetic detective. So I hope that you can feel the same way. We'll find out shortly. But with the tool, again, you can enter in different types of media and create bookmarks. So I try not to use so many bookmarks in my Chrome browser anymore. I try to track the papers through this tool and so on. So this is just what it'll look like if you do a search and you pick out a paper, and then you add a bookmark to it, which is basically just like a label, and then you can see all the papers that you have bookmarked with metagenomics or some other term and the title. And the numbers here tell you how many, it's not color separated, but how many annotations were extracted from the PubMed file, and then how many were manually added. So now is not so many. And then you can go in and you can edit those. You can see if there's certain papers that you're interested in that when you have time you want to go back. We always start with our own things first. So I know you're still all a bit young, but maybe you have some particular area that you're interested in, and that might be informative for you to look through certain types of papers, maybe add some information. Again, for your own benefit, because you're doing some kind of research, but then also for the crowd's benefit, that by adding this additional information, it'll help other people find that easier than you did. And we can keep track of our searches if we want to go back and see, are there new papers in my field, or if I use these certain search terms, have any new ones come out since the last time I searched. We can make user communities and collaborate. So all of the data that goes into our tool is, just like with Wikipedia, it's freely accessible to anybody. So again, if I have this kind of top secret idea I'm working on, I'm not going to put that here. But if I'm just extracting the information about the genes, the authors, the figures and things in the paper, I can share that with people. Let's say we're all working on the mouse genome together, and we've identified a bunch of papers that are new, and we want to go through those papers as a group and try to extract certain information. So we can create our own community, we can share the bookmarks within the group, and we can also share the vocabularies that we want to add to each of those papers. We can have a private group, a public group, but the data itself will become public. And once we have the data there, we can start to visualize it in different ways. And so in our app, we actually want people to contribute. So again, the way I want to look at the information might be completely different than the way that you would want to look at it. So you can say, oh, Todd, but could I create a graph that would show me the history of all the papers in my area over the years? So we could actually, by doing some scripting, we can actually create a visualization for the question that you're trying to ask, and then we can share that with people. So, for example, like this, I think I have a couple of close-ups. This one shows the different media types. So these are journal articles, and I can't see here, audio files, and YouTube, or videos and images and so on. And the number of people that have contributed the annotations, that shows how many annotations were contributed. We can see which are the current terms that are most commonly being added to papers and so on. This is just another way, very complex here, to see how everything becomes interconnected with everything else. But we can use this kind of information to maybe suggest. So I see that you've bookmarked certain types of papers. So just like Google does or Gmail, if you use that, they suggest you different products based on your Gmails or maybe your shopping habits or so on, and then suggest you, hey, you may also like this. So because you like these papers, you might also be interested in these new ones. So this isn't a reference manager. I don't know if you've ever used like a reference manager tool. If you're writing a paper, it helps you keep track of all your references, automatically like inserts them into your document. That's always a complicated part of when you write a paper. It's not a reference manager, but maybe it could be. And it's not a place to store the media. So when you can view your paper in our tool, but it's not actually being sent to our server. It's just only visible on your local computer. We're not storing those, and we're not storing YouTube videos and things like that, even though you can see them when you're in the tool, because we don't want to be violating copyright, and we don't want to be paying for like infinite amounts of storage either. So I have text mining, web mining, data mining. Our tool is not a mined resource. So each one of these has its using some computer algorithms to scroll through, sort through papers and try to extract information. But there's like a certain amount of reliability that until a human has actually checked that information, we should still be somewhat skeptical. So we are dependent on our tool we want to have like the most reliable information going into the database. You guys are all first timers, and I'm going to be asking you to add some information into the database. So you're saying, oh, do I need to be an expert, you know? And then actually, no. But the crowd can also give feedback to itself. So if I'm going through the database and I see somebody added this annotation about my paper and I know that's wrong, I can like vote it down. So we can start to pick up patterns, and then maybe I can maybe contact you and say, hey, what were you trying to, I think maybe you've misinterpreted the results here. What were the information you were looking for? So the more reliable the information is in the database, the more reliable we can do our searches and things. So, why should you care or contribute? Okay, so you can keep track of media that you're interested in. Again, share your non-secret thoughts so that everybody can take advantage of that information and save other people from wasting their time by trying to look for the same thing in the same paper hundreds of times over again. I already mentioned those. Search exactly what you're looking for, make your own work more discoverable, and then uncover connections between research or between media that maybe you didn't know were there. Websites and URLs can sometimes die. So at one point we know these things existed, but now that resource is no longer available or the page has changed. If we happen to capture it in our database, it will stay there, not the full web page, but at least the information describing that page. And then media can stagnate. So PubMed, once something goes in there, it just stays that way. So the problem is that new terminologies come along. So like the word metagenomics has only really been popularized maybe in the past 10, maybe 15 years if you stretch it back. But before that, there were papers or research going on in that field, but they didn't have that term. So, but because that was put in the database a long time ago, nobody ever goes back and adds on the new terminology. Another thing they recently is becoming popular in science is what's called like a researcher ID. So the problem is we have many people, just based on a person's name is not enough to say, this June Wang and this June Wang. How do I know? They use the same Chinese characters. How do I know which is which? I had one example, not showing it today, but where you had this one really large paper, you had 32 people named like J. Wang. And how do you know? You want to tell your mother, hey mom, I published this paper on something. She, oh, which one are you? Oh, I'm number 324 out of 2,000 authors. What often happens in some societies when you get married, then the person's name might change. So if you publish some of your first papers with your pre-wedded name, and then you get married, you change your name, you don't want to disconnect your work with your new name from your previous name. But by using this researcher ID, it doesn't matter what your name is. So this is something that just came along in the past couple years. I'd like to go back and add that my researcher ID to all of my previous publications. So with our tool, we can do that. Doesn't matter how old the data is. We can always go back and we can add new terminologies, new information. Ideally, what we'd like to do is feed that back to PubMed. Hey, why not take this information and include it in your database? So I mentioned many of them are non-English. Make your own book. I think most of these things I already mentioned. And then get community feedback to improve the annotations. So the same thing like with Wikipedia, any kind of crowdsourcing tool, there's usually some managers or people that are, once you've been doing it long enough, you become an administrator, you can either edit other people's work or make suggestions to it or give them some kind of feedback. So that's how we can improve the information that goes in there. And try to push to make more media become open source, because a lot of the articles in research are still, we have to pay in order to see the full paper. We're hoping by doing something like this, we can encourage and sending more people to these journals that might encourage them to make their papers more easily accessible. So what I'd like you to do, this kind of just shows a map of all the people who have registered with our tool in the past, is now to get out your laptops and then let's all give it a try. So just kind of boring shapes, maybe.
It's still not valid.
In our previous one, we wanted to let people like they're both talking about the same thing. And we have this big scope. Actually, we can finish on the vocabularies that I was talking about earlier.
That's definitely heads up. You know what?
So that was like, so